introduction to speak to you for 8 minutes about innovation in Africa. And I think it rounds off the discussions that you've already been engaging with here this morning. The last time I actually came to Ethiopia, and I think it was in this very room, I came to address the African Union plenary on the importance of science and technology and innovation in Africa when I collected the Kwame Nkrumah Prize. And I was struck at the level of engagement I saw at that level. So what I'm going to talk to you now is about one example. And I'm really just a spokesperson for the many great achievements in science in Africa. And I'm hoping that I will convey to you just one way in which Africa is contributing to dealing with the HIV epidemic. Now, UNAIDS estimates that there are about 1.8 million new infections that occurred last year. That translates into about 5,000 new HIV cases each and every day. So to people who tell me, you know, we've solved this problem, we've turned the corner, well, let me tell you, no corner's been turned if you're getting 5,000 new cases of an infection each and every day. Africa, that bears 70% of the global burden of HIV, bears the brunt. And within Africa, young women have the highest rates of HIV infection. Indeed, if you look at the global target that we set at the United Nations, the target for 2030, we can simply not achieve that if we do not make a dent on reducing the high incidence rates of HIV infection in young women in Africa. Young women in Africa present to us one of the biggest challenges in dealing with the global HIV epidemic. Now, we've been involved with many other scientists in trying to find solutions. In 2010, we announced to the world that antiretrovirals can be protective against HIV. Shortly thereafter, a few years later, the World Health Organization recommended the use of tenofovir-containing antiretroviral regimens to a tablet a day to protect you from getting HIV. We've had many challenges in implementing that. And that intervention called PrEP, pre-exposure prophylaxis, has not yet had the kind of impact we would like to see, because it's really difficult to take a tablet every day to protect yourself against HIV. So when we look now at new solutions and how we might find new solutions, we've been looking for ways in which we can empower women to choose a technology that they can use that fits into their lifestyle, that they don't have to deal with the daily challenge of taking a tablet. And so one of the things we've been trying to look at is what long-acting options are there. Let me switch now and come back to this issue of what long-acting options there are. Every person, when they acquire HIV, anyone acquiring HIV, the body naturally responds by creating antibodies. You remember this from high school biology. Now, normally, when a person responds to an HIV infection, the antibody is highly specific to that particular infecting virus. The antibody that is made by the human only will kill that particular HIV. It was discovered in about 2010, 2011 by two groups in the US that there are these rare individuals who make something called broadly neutralizing antibodies. These are antibodies that can kill a whole range of different kinds of HIV. And HIV is highly diverse across the globe in different subtypes and different ways in which they present. So when we realize that there are these individuals, in one of the studies that we've been looking at acute infection, we screened thousands of our women participants. And in screening them, we discovered that there's one woman Her code name is CAPRISA 256, or CAP 256 for short. And when we screened her serum against a panel of 400 viruses from throughout the world, viruses from throughout Africa, Australia, from Europe, from South America, from all over the world, we found that her antibody could kill about 4 out of every 5 of those viruses. So we discovered that she has a broadly neutralizing antibody. But there was something unusual about it as we were doing the experiments in the lab. There was something unusual about the way in which the antibody was having an impact in neutralizing HIV in the test tubes. So we worked with our colleagues and we said, we've got to find a way to develop this. We've got to find a way to really work with this antibody. And so we sent a student, a PhD student, to go off to the NIH to work with John Mascola and Nicole Doria-Rose at the Vaccine Research Center and to see if they can find the cell, the B cell that makes this antibody. Now you remember, from high school biology, B cells and T cells. Among the B cells, you got one particular antibody is made by about 1 in 10,000 B cells. Just think about that. 1 in 10,000 B cells, on average, makes an antibody. So when we sent the student with a vial of serum and said to her, go and find that cell, we knew it was a needle in a haystack. We knew the chances of finding it were really going to be a challenge. We told her, go and spend 3 months, see what you can get out of this, and at worst, you'll come back and we'll be able to work it here. Well, just before she left, I got that call. I have found the cell. That one cell that makes this particular antibody. Using that cell, we cloned it in the lab. We then made the first 3 grams of the antibody. We sent it off to our colleague Dan Baruch at Harvard University. He put it into the monkeys and challenged them, and that's when we realized what we were dealing with. 9 monkeys that received the sham injection all acquired monkey HIV. The 12 monkeys that received CAPRISA 256 antibodies, not one acquired HIV, even at the lowest doses. We now knew we had something that works in animals. Next step, we've got to put it into humans. So armed with this data, we go off to the NIH to see Dr. Fauci and Dr. Mascola, and we say to them, we've got to manufacture this at GMP level, we've got to put it into humans. And so we've been doing that. We've spent the last 2.5 years working with the VRC to manufacture this antibody. 3 weeks ago, they sent me the photograph of the vial with the antibody. We finally have the antibody. And we are about to start injecting the first women in South Africa with this antibody. The point I want to make here is that here we have 3 things that I want to share. The first is that we can find solutions to the HIV epidemic right here in Africa, where 70% of the burden exists. So it is possible to develop and be innovative right here in Africa. The second is we could never have done this without the partnerships. We have about 300 different researchers across 4 continents working on this project with us, all led by African scientists. And finally, we could never have done this without many of the partners coming together with the funding. And here, a study like this costing in the region of around $40 to $50 million. The NIH picking up about 1/3 of the tab, the European Union picking up 1/3 of the tab, and very importantly, the South African government, through the Department of Health, through the Department of Science and Technology, and through the South African Medical Research Council, is picking up about 1/3 of the tab. That's when we talk about local ownership. And that's what I was listening to when I heard Francis Collins and CD talking about this taking charge and owning it here. So I hope to tell you when we go to the next grand challenges of what the results are from the clinical trials that we are undertaking with this antibody. Until then, watch this space. Thank you.